Multiplexed assays in droplet microfluidics represent a revolutionary approach that combines the efficiency of high-throughput droplet generation with the power of simultaneous multi-stemmer print analysis. Imagine a massive laboratory where millions of tiny test tubes operate in parallel, each performing a unique assay under identical, tightly controlled conditions. In this system, a sample is partitioned into thousands or even millions of microdroplets, each droplet acting as an independent reaction vessel. Within these droplets, researchers can perform different biochemical reactions or detect multiple analytes simultaneously. This is similar to having a miniaturized supermarket checkout where every item is scanned at the same time rather than one after the other, saving both time and resources while providing comprehensive data from a single sample. At the heart of multiplexed assays in droplet microfluidics is the precise control over droplet generation. Microfluidic chips are engineered with intricate networks of channels where immiscible fluids, usually an aqueous phase containing reagents and an oil phase that stabilizes the droplets, meet at carefully designed junctions. Techniques such as flow focusing or TJ junction formation create droplets that are nearly identical in size and composition. Think of it as a highly advanced cookie cutter that produces perfectly uniform cookies every time. Uniform droplets ensure that every assay within each droplet is performed under the same reaction conditions, which is critical for reliable quantitative measurements. To illustrate, imagine a clinical diagnostic application where a patient's blood sample is analyzed for a panel of biomarkers, say a dozen different proteins or genetic markers associated with a disease. In a multiplex droplet microfluidic system, the blood is first mixed with various reagents, and then the mixture is segmented into thousands of droplets. Each droplet is pre-programmed with a specific set of capture antibodies or nucleic acid probes that target one particular biomarker. As the droplets flow through the microfluidic chip, they are incubated under controlled conditions and their fluorescence or colorimetric signals are recorded. This is like having a series of mini laboratories working concurrently, each one testing a different hypothesis about the patient's health. A common question here might be, how does this compare to traditional ELISA or PCR tests? Traditional methods require separate reactions for each target, often using milliliter volumes and taking hours or days. In contrast, droplet microfluidics can perform thousands of assays in parallel with nanoliter volumes, dramatically reducing time, cost, and sample consumption while increasing sensitivity and throughput. The science behind multiplex assays involves several key components. First, the precise control over reagent mixing is essential. In a droplet, reagents mix primarily by diffusion, a process that, while slower than turbulent mixing, ensures that the reaction conditions remain uniform across the droplet. This predictability is crucial for obtaining reproducible results. For example, different fluorescent labels can be assigned to different targets, allowing a single droplet to be read in multiple channels. Imagine a rainbow where each color represents a different biomarker. When a droplet glows under the microscope, the pattern of colors reveals the specific combination of biomarkers present. You might ask, how do researchers prevent overlap or interference between signals? The answer lies in careful selection of fluorophores with non-overlapping emission spectra, and the use of sophisticated optical filters and detectors that can separate and quantify each signal accurately. Moreover, microfluidic platforms often incorporate advanced techniques such as pico-injection, where additional reagents are introduced into pre-formed droplets. This capability is particularly useful in multiplexed assays because it allows for multi-step reactions within a single droplet. For instance, a droplet might first contain a sample and a binding reagent, and later a washing or amplification step can be performed by injecting another reagent into the same droplet. This sequential processing is akin to cooking a multi-course meal in one small pot. Each ingredient is added at just the right time to achieve the desired final result. Analogies help make these concepts more tangible. Picture a multiplexed droplet microfluidic system as a high-tech automated kitchen where every dish or assay is prepared simultaneously in its own tiny, perfectly controlled chamber. Just as a gourmet restaurant might serve multiple courses that each require different cooking techniques and precise timing, these micro-droplet systems conduct various biochemical reactions concurrently with remarkable precision. Each droplet is like a mini chef's workstation, complete with its own set of ingredients, cooking time, and temperature settings. When you combine all these mini workstations, you get a full, comprehensive banquet of data that can be analyzed to provide insights into complex biological or chemical processes. Another compelling application is in single-cell analysis. In this context, cells are encapsulated in droplets along with a panel of reagents to study their gene expression, protein levels, or metabolic activities. Each droplet acts as an isolated environment where the cell's behavior can be examined without interference from neighboring cells. For example, researchers have used droplet-based single-cell RNA sequencing to profile the transcriptomes of thousands of individual cells from a tumor sample. The resulting data reveal the heterogeneity within the tumor, helping to identify subpopulations of cells that might be responsible for drug resistance or metastasis. You might ask, how is such detailed information obtained from such small volume? The answer is that by partitioning the sample into thousands of tiny droplets, the concentration of each cell's biomolecules is effectively increased relative to the droplet volume, making detection by sensitive optical or sequencing methods feasible. In environmental monitoring, multiplexed droplet assays can be used to detect a wide range of contaminants simultaneously. Imagine a water quality test where each droplet is designed to react with a different pollutant such as heavy metals, pesticides, or microbial toxins. As the droplets pass through a detection array, they might change color or emit a fluorescent signal in the presence of their target contaminant. This allows for rapid on-site analysis of water quality, which is critical in scenarios such as disaster response or routine environmental surveillance. The parallel processing capability of droplet microfluidics ensures that even low concentrations of contaminants can be detected quickly and reliably. Data management in multiplexed assays is another fascinating aspect. With thousands or even millions of droplets being analyzed simultaneously, the resulting data sets are enormous. Advanced computational methods, including machine learning and artificial intelligence, are used to process and interpret the data. Algorithms sift through the optical signals, identify patterns, and quantify the presence of various targets with high precision. This high-throughput analysis is like having a supercomputer as your sous chef, rapidly sorting through countless recipes to determine exactly which ingredients yield the best flavor profile. A common question that arises is, what are the main challenges in multiplexing assays and droplet microfluidics? One significant challenge is maintaining droplet stability. Droplets must remain isolated and uniform in size throughout the assay, and surfactants are used to prevent droplet coalescence. Optimizing the concentration and type of surfactant is critical because any droplet merging can lead to cross-contamination and erroneous results. Another challenge is ensuring that each droplet is accurately labeled and sorted, especially when dealing with complex mixtures. Innovations in fluorescence-activated droplet sorting, FAD, and high-resolution imaging are addressing these challenges by providing robust systems to identify and separate droplets based on their unique optical signatures. Looking ahead, the future of multiplexed assays in droplet microfluidics is extremely promising. Researchers are developing integrated lab-on-a-chip systems that combine sample preparation, droplet generation, multiplex detection, and data analysis into a single, compact device. Such platforms have the potential to revolutionize point-of-care diagnostics, where a small patient sample can be processed and analyzed in minutes, yielding a comprehensive panel of diagnostic information. In drug discovery, multiplex droplet assays can screen large libraries of compounds against multiple targets simultaneously, significantly accelerating the identification of promising therapeutic candidates. To sum up, multiplexed assays in droplet microfluidics provide a powerful, high-throughput method for analyzing multiple biochemical reactions concurrently. By dividing a sample into uniform microdroplets and precisely controlling the reaction conditions within each droplet, scientists can perform simultaneous assays with remarkable sensitivity and specificity. Whether it's for clinical diagnostics, environmental monitoring, single-cell analysis, or drug discovery, the ability to conduct multiplexed assays on a microfluidic platform is transforming how complex analyses are performed.